fine tunes it. Let's see, to introduce this question, let me remind you that we saw that in the lecture before last that tubuloglomerular feedback works by the macula densa cells analyzing the sodium load coming through the distal convoluted tubule in the nascent urine. The distal tubule folds back in the nephron to pass right by the same glomerular arch where the afferent arteriole comes in on one side and the efferent arteriole goes out on the other. The macula densa is right by those two arterioles and forms the juxtaglomerular apparatus. Those three things are the juxtaglomerular apparatus. We saw last time that the juxtaglomerular apparatus can instruct the afferent arteriole to constrict. That's a direct message through adenosine, as well as ATP, which gets converted extracellularly to adenosine, sending like a text message to the smooth muscle in the afferent arteriole saying constrict. And that you should also be reminded while we're at it that the stretch receptors in the smooth muscle cells also contribute to that automatic constriction when blood pressure is high to pull glomerular filtration rate back down again. So that's a feedback mechanism. Today we're going to talk about we're going to start with another mechanism carried out by the juxtaglomerular apparatus, this time when the sodium load is low. And so question number one is, which enzyme is released by juxtaglomerular cells of the kidney and cleaves angiotensinogen made by the liver? Which enzyme converts angiotensin 1 to angiotensin 2? What now famous virus binds to said enzyme? And what effectors mediate the angiotensin 2 signal? And to what end? Go. Ian? We're learning the details about the RAAS, renin-angiotensin aldosterone system. Renin-angiotensin aldosterone system involves a hormone named for coming from the kidney. The reins are the kidneys. Renal blood flow is kidney blood flow. Renin is a hormone coming, well, it's really an enzyme being secreted by the juxtaglomerular apparatus when the macula densa cells signal it by secreting prostaglandin. Prostaglandin stimulates renin release. And the renin or renin, either pronunciation is correct, is an enzyme that gets into the blood, cleaves angiotensinogen into angiotensin 1. Angiotensin 1 flows throughout the bloodstream, finding angiotensin converting enzyme on the surfaces of the endothelial cells. And that converts angiotensin 1 into angiotensin 2. Angiotensin 2 primarily activates the adrenal cortex to stimulate it to release more aldosterone. And we saw last time that aldosterone increase sodium reabsorption in the distal convoluted tubule. It's a specialized final touch that allows the kidneys to help maintain the osmolarity of the blood, the sodium concentration, particularly of the blood. But angiotensin II also does other things that we're learning for the first time in this session. It stimulates the hypothalamus to release vasopressin and stimulates the thirst center in the hypothalamus, so we drink more water. It stimulates the CBCC to increase blood pressure with increased contractility of the heart and heart rate. It stimulates the arterioles to constrict, propping up the central blood pressure. So there's not as much, I want to call it, flow out of the central arteries, and they have a maintained pressure. So all of those things end up improving the blood pressure when it was low in the first place. You can see my cursor, can't you? And that's what prompted the renin release. Interesting, no? There's baroreceptors, the renin, and the renin helps with angiotensin, and the angiotensin causes arteriolar constriction to prop it back up, as well as stimulating the CBCC directly, the hypothalamus directly. Vasopressin, what does that do? That stimulates water reabsorption through aquaporin insertion in the collecting ducts. Question 2. When blood pressure is too high, the glomerular filtration rate would increase. Were it not for tubuloglomerular feedback, where adenosine released from the macula densa stimulates afferent arteriole constriction. But when blood pressure is low, what does the macula densa secrete, and how does it increase blood pressure, complementing the baroreceptor reflex? Okay, everybody's in, Morgan tells me. So the answer is that the macula densa releases the paracrine prostaglandin when blood pressure is low. And that stimulates the release of renin to do all those things we just talked about. In this slide, the decreased blood pressure is effect is emphasized a bit by pointing out that paracrines, prostaglandin, cause renin secretion. The low blood pressure would cause renin secretion. And also, here's a new one. Sympathetic nerves actually stimulate the juxtaglomerular cells and stimulate the renin secretion. So if the blood pressure is down, not only does the CBCC tell the heart to speed up, it tells the kidney to speed up. Interesting, hey? So that complements the heart effect of the CBCC. Remember, the CBCC is in the medulla. It sends output to the locus coeruleus, which is the head of the sympathetic system. And the sympathetic nerves travel down through the lateral horns, or to the lateral horns, where they send preganglionic nerves out to the sympathetic chain. And those can go to the kidney. Next question. If you eat a bag of salty potato chips, your blood osmolarity, sodium chloride concentration in the plasma, will go up. You need to dilute the salt. How do your hypothalamus and kidneys work together to accomplish this? Go. Sodium chloride doesn't appreciably increase the volume of the blood as it's absorbed through the stomach and intestines. It's pure salt, and that actually contracts the water around it to decrease the volume a little bit. But the osmolarity has gone up, and the osmolarity center in the hypothalamus recognizes that there are sensors that stimulate the posterior pituitary gland to secrete vasopressin and stimulate the sensation of thirst. There's a thirst center in the hypothalamus that makes us want to drink more water, to balance out the excess sodium that we've taken in. Now that extra water might return the osmolarity to normal, but it's going to increase the blood volume when we do that. So we'll get a headache, at least I do. So then we have to hope that the blood pressure reflexes kick in to bring the blood pressure back down to normal. First, the rapid response through the CVCC and reduction in the sympathetic outflow of the locus coeruleus. But also the impact on secretion of renin, and the antidiuretic hormone, stimulating the kidneys to conserve water, the aldosterone secreted by the adrenal cortex under the stimulus of angiotensin II, stimulating increased secretion of salt and water. Boy, let's see, I have that backwards, I'm sorry. There would be a decrease in aldosterone for lack of renin. I set it upside down. And that would slowly cause less and less sodium reabsorption, and therefore more and more excretion of salt and water in the urine. Okay, next question. If you drink too much water, not soda, pop, water, not only do your atrial stretch receptors signal the hypothalamus by nerves that the blood volume is too high, but also there are endocrine cells in the atrial wall that release ANP, atrial natriuretic peptide. Now, ANP is a hormone. It's a peptide hormone that brings about multiple effects. Name 3 target tissues for the hormone ANP that increase renal excretion, and name the one in the medulla oblongata that reduces sympathetic tone.
1 second. Apparently no one can see question 4. So I'm trying to get into it and see if it's open for them or what's going on.
Can I leave that to you?
Yeah, go on.
Morgan says he's working on opening question 4 for you. I must have goofed.
323 open at 9 a.m.
Does the participants list say 26? I thought it did.
Students, edit participants. It's this one, right? The antidiuretic hormone.
No, this is not the one I just read.
Oh, that's 25. Oh, maybe that's the participant.
Oh, it says all students. Maybe
Yeah,
the schedule isn't right. You have to go to top line of digital discussions. Click on it.
Okay.
Not the question itself. Go to.
Can't you do it like right here? Go to on this side. Let me see. Oh, yep. Here we go.
Okay.
Give
I put it
2
as
seconds and it
PM
should be
instead of AM. Sorry.
there. Okay, it should be there if you
Now it
reload.
should be there. Reload your screen. Sorry, my fault. If you drink too much water, you get ANP released. What are 3 target tissues that increase renal excretion and 1 that reduces sympathetic tone? Go. Oh, did you remember to record this session? Wow, we're all together. Thank you. So the atrial natriuretic peptide influences the kidney, the hypothalamus, and the adrenal cortex. Let's look at how it does those 3 things. The hypothalamus takes the ANP coming from the atrium when it's stretched by the increased volume and says, we don't need to reabsorb water, so let's quit secreting vasopressin, which is ADH. The kidney receives the hormone in its receptors, and the juxtaglomerular apparatus is told not to put out so much renin because we don't want to reabsorb sodium or cause vasoconstriction through the angiotensin aldosterone mechanism. But the kidney also will have an increased glomerular filtration rate because the extra volume will result in an increased blood pressure, and that will stimulate the filtration at the glomerulus. And it has direct sodium chloride and water excretion changes as well. The adrenal cortex will have less aldosterone, as we said, because there's less renin. So ANP affects the kidney directly, the hypothalamus, and the adrenal cortex to stimulate sodium chloride excretion if we have too much blood volume. It also, not just through the baroreceptor reflex, but directly through ANP binding in the medulla oblongata, the CBCC, also inhibits the sympathetic outflow so that you get a decrease in blood pressure to compensate for this water drinking binge you were on. I wonder. That was question 4, right? Yes. I was looking at question 3. So maybe you were all in, maybe you weren't. Question 5. Many of you are in already. How might you decrease blood volume while increasing blood osmolarity? What mechanisms would reverse that derangement? Several good answers. This is kind of the Punnett square of derangements of homeostasis of volume and osmolarity. It's got 9 boxes, notice. The central one is normal. All of the surrounding boxes are derangements. What happens when you get out of homeostatic normality? And how do you do it? Well, the question started out, how do you get this one? Increased, wait a minute, where am I? This one. Increased osmolarity, but decreased volume. Increased osmolarity, decreased volume. Dehydration is the basic mechanism that results in that derangement where you've sweat, hypotonic saline. What that means is your sweat is salt water, but it has less salt than your normal plasma has, because some of this salt coming from the plasma in the sweat glands gets reabsorbed, so you get more pure water. And that's good because the pure water evaporates better and cools you better. So sweat is typically hyposmotic, hypotonic, and that results in an increase in osmolarity of the blood. You've reabsorbed the sodium from the sweat, not the water, so you've got excess sodium in the blood. But at the same time, you've gotten rid of water, so your volume has decreased. So you've gotten dehydrated. Maybe you've run, say, 15 or 20 miles, and you've gotten dehydrated. In order to restore normality, you're going to have to increase your volume and decrease your osmolarity. How can you do both of those things? Well, in the case of dehydration, a lot of different mechanisms come into play. The low volume stimulates the CVCC to increase the heart rate, increase the cardiac output. That brings the blood pressure up to normal in spite of the low volume. Also, the arterioles vasoconstrict because neurons from the sympathetic chain stimulate them to constrict. Furthermore, renin cleaves angiotensinogen. ACE cleaves that into angiotensin II, and that stimulates the arterioles to constrict. The glomerular filtration rate's down because the blood pressure is down, so there's not as much filtration force. Starling's law of the capillary says you won't get as much extravasation, which helps automatically because if you filter less, you lose less to excretion. So that will bring your blood volume back up to normal. The tubular-glomerular feedback will do the same thing through the juxtaglomerular cells. Angiotensin I would stimulate the adrenal cortex to release aldosterone. So that should go up, but there's a down arrow right here. Odd. Well, notice that the adrenal cortex has two blue stops here, meaning it's actually not stimulated to secrete aldosterone. Here's one of those exceptions that complicates matters. That it's a fine point, you just have to know that the adrenal cortex also has osmolarity, detectors in it. And the increased osmolarity inhibits it in spite of the stimulation from angiotensin II, so it doesn't put out aldosterone after all. That's a good thing because you don't really want sodium reabsorption. You have too high of an osmolarity. And so you need to cut down on sodium reabsorption so that you have more sodium excretion, reducing your blood osmolarity. Finally, the vasopressin mechanism will stimulate water reabsorption to bring your blood volume back up, but it's pure water, so it won't bring your osmolarity higher. In fact, it will dilute your osmolarity, bringing it down. All of those are great things. Question 6. Through metabolism, our body has an acid load. There's hydrogen released from bicarbonate, from carbonic acid, when CO2 is converted to carbonic acid, and that turns the carbonic acid into bicarbonate. Lactic acid from sugars in glycolysis, fatty acids and keto acids from beta oxidation of fats. All of these things create an acid load, a constant acid load. The proteins and bicarbonate in the plasma buffer the plasma pH so that it stays near 7.4. But the lungs and kidneys have to excrete the acid as fast as it is produced, or the buffers would eventually get overwhelmed, How do the lungs excrete acid? Go. We breathe off CO2. If we have more acid in our blood, we'll breathe harder to get more CO2 out. That leaves the process of bicarbonate binding hydrogen and then breaking in half to turn into water and CO2, reducing our free hydrogen concentration. Free hydrogen concentration is related to the pH. As the concentration goes up, the pH goes down. Why is that? Because pH is minus the log of the free hydrogen concentration. So the Henderson-Hasselbalch equation has importance in maintaining the free hydrogen concentration in the context of CO2. So we breathe out our CO2. That reduces our acid load, but you can only reduce the acid load so much that way. And maybe you can't breathe harder because you have asthma or you have coronavirus pneumonia, and so you're not really getting rid of the CO2 from your lungs. What do we call that? What's the failure of the lungs to excrete enough acid in the form of CO2? What's that metabolic derangement called? It's called respiratory acidosis. Acidosis because you get acidic blood. Respiratory acidosis involves having high carbon dioxide in the blood, which then leads to high bicarbonate as the CO2 combines with water in the Henderson-Hasselbalch reaction to make carbonic acid. And that then dissociates, one of the hydrogens falls off easily to cause low pH, high hydrogen concentration in the plasma. Asthmatics would have a risk of having respiratory acidosis. Acidosis means acidic blood. Respiratory because it comes from failure to eliminate CO2 through respiration. You're going to need to have a good solid understanding of all of these four derangement squares. Acidosis, too low a pH. Alkalosis, too high a pH. Respiratory because of breathing. Let's talk now finally about how the kidneys also play a role in excreting acid and fine-tuning the pH of the blood. Next question. Question 8. How do the kidneys excrete the acid load on a steady, ongoing, the sophisticated word is constitutive, that's a steady, ongoing basis, producing a generally acidic urine? Go. Come back to that in just a second. No, let's start with that. The answer is that the I do want to start with that. That phosphate and acidic urine, free hydrogen, are excreted in the urine. Also, ammonium is excreted through pumps in the apical membrane. So ammonium, that's that nasty smell of baby's diaper. Phosphate, they carry the hydrogen out. HPO4 2- picks up a proton that's secreted at the apical membrane, and generally bicarbonate is reabsorbed at the basolateral membrane through transporters that exchange bicarbonate for chloride. This takes place, you might think of it primarily as the proximal convoluted tubule, or throughout the whole tubule, kind of like the sodium reabsorption and glucose reabsorption, also amino acid reabsorption that we didn't talk about much. All of those take place at the beginning, proximal convoluted tubule the most, and they kind of set the stage. They say that they compensate for the acid load. Last question. The pH of the urine, if fine-tuned by cells in the, is fine-tuned by cells in the collecting duct called intercalated cells. The type A intercalated cells excrete acid, while the type B intercalated cells excrete base. What transporters in the apical and basolateral membranes and enzyme in the cytoplasm are responsible for this function? And how are type A cells different from type B cells? Okay, the answer is that the type A cells have proton pumps in their apical membranes to excrete protons, into the lumen of the collecting duct. They're found in the CD, the collecting duct. They're intercalated between other types of cells. So some are type A, some are type B. There's other collecting duct cells as well. Distributed. And the A's and B's are opposite from each other. The A's have these proton pumps in their apical membrane, but the B's have proton pumps in their basolateral membrane. So as the proteins are synthesized through the Golgi, they have to be trafficked to the right membrane. The proton pumps can be proton ATPase or proton hydrogen potassium antiporters, both active transporters. If they're in the basolateral membrane, as in B basolateral, never thought of that before, then they are set up to pump protons out into the interstitial space. If they're in the apical membrane, A, never thought of that either, they are positioned to pump protons the opposite direction, out into the lumen from the cytoplasm. So in both cases, they pump protons out, but if they're in the apical membrane, it ends up in the lumen. If they're in the basolateral membrane, the protons end up in the interstitial space. Now, they don't work alone. There's also a need for chloride bicarbonate exchangers to relieve the high buildup of bicarbonate in the cytoplasm, letting chloride in to take its place. That's either in the basolateral membrane in the type A cells or in the apical membrane in the type B cells. Notice that this would have the effect of excreting bicarbonate. Think of bicarbonate as a base. The bicarbonate is being excreted in alkalosis when the extracellular fluid is too low in hydrogen, too high in bicarbonate. In both cells, carbonic anhydrase is crucial to convert CO2 to bicarbonate. But what happens to the bicarbonate depends on which cell type it is, which depends on, and therefore on what pumps it has in the membranes. That's it for today. Let me back up.
Question real quick before we move on. Someone said, nope, sorry, gotta do this one. Sorry. How do type A and B cells get different signals so they don't just cancel each other out?
The type A cells respond by, when the hydrogen is high, by pumping hydrogen out. And the type B cells don't do anything. The type B cells respond when hydrogen is low by pumping bicarbonate out, and the type A cells don't do anything. Because type A cells depend on this high hydrogen to fuel their proton excretion. And the type B cells depend on this bicarbonate and the hydrogen gradient to fuel their bicarbonate excretion. That's a good question. Probably not completely answered, but at least that's the starting point. Let me also back up to this slide that we kind of skipped over, just to remind you that it's CO2 affecting the central chemoreceptors through diffusing through the blood-brain barrier, then through Henderson-Hasselbalch producing hydrogen, which actually is what binds to the central chemoreceptors to stimulate respiration, to make us breathe harder, to get rid of CO2 when we're compensating for high CO2. By the same token, if we're compensating for low blood pH, we'll do the same thing. The peripheral chemoreceptors in the carotid bodies and the aortic bodies that are near the arch of the aorta send sensory neurons to cause us to breathe higher. So this was the original respiratory response to the derangement. The metabolic response to the derangement will be in the kidney primarily, affecting the pH and the bicarbonate levels. But don't get mixed up here. It's the kidney responding more long-term and steady to a metabolic acidosis or alkalosis that we're really referring to. And the respiratory response is rapid for either metabolic or a respiratory acidosis. The respiratory response happens in minutes, the metabolic response over the course of hours. That's it for today. Thank you for coming. One last question
is, respiratory acidosis is also hypoventilation, is that correct?
Hypoventilation also causes respiratory acidosis, yes.
Okay, I think we're good.
Thanks, everybody. Congratulations on sticking to it. Hang in there. Keep getting your points, doing your homeworks, reading the book, and we'll have a decision on the final exam pretty soon now. Take care.